Hello all, welcome to this second part of plant physiology and plant anatomy video. In exams, Vidhai is a non-profit initiative by the students of IIITDM Kancheepuram to give free coaching and resources to the students who are preparing for national level competitive exams like NTSE and NMMS. So in this second part, we'll be looking into the internal structure of the leaf and then the differences between monocot and dicot leaves. Then finally, we'll be diving into plant physiology and then different organelles of the plant, of a cell, plant cell. So coming to the internal structure of the dicot leaf, the transverse section of the dicot leaf is shown. The first layer is the upper epidermis, this is the outermost layer of the outermost layer made up of single layered parenchymatous cells without any intercellular spaces, which you can observe in the diagrams is shown. And then the outer wall of the cells are cuticularized. Stomata are less in number. Then comes the lower epidermis. It's a single layer of parenchymatous cell with a thin cuticle. Contains numerous stomata. Chloroplasts are present only in the guard cells. The lower epidermis helps in exchange of gases. The loss of water vapor is facilitated through this chamber. And then we have the mesophyll. This tissue is present between the upper and the lower epidermis. It is differentiated into Palisade Parenchyma and spongy parenchyma. So the Palisade Parenchyma is found just below the upper epidermis. These cells are elongated. The cells have more number of chloroplasts. The cells do not have intercellular spaces and they take part in photosynthesis. Now coming to spongy parenchyma, it is found below the Palisade parenchyma tissues. These cells are almost spherical or oval and are irregularly arranged. These cells have intercellular spaces and they also help in gaseous exchange. Now coming to the last part of the leaf, which is the vascular bundle. The vascular bundle is the bundle of a midrib is larger, which is like in the leaf we'll have the midrib, from which the branches will arrive. If we can take a leaf and can see that. The vascular bundles are conjoint and collateral. They are closed. Each vascular bundle is surrounded by a sheath of parenchymatous cells called the bundle sheath. Each vascular bundle consists of a xylem lying towards the upper epidermis and a phloem lying towards the lower epidermis. So this is the structure of to brief about the internal structure of a dicot leaf. Now coming to the internal structure of the monocot leaf. Here again the transverse section is shown for reference. The first layer is the epidermis. The monocot leaf has an upper epidermis and a lower epidermis. The epidermis is made up of parenchyma cells and cuticle is present on the outer wall. Stomata are present on both the upper and lower epidermis. Some cells of the upper epidermis are large and they have thin walls. They are known as bulliform cells. Next layer is the mesophyll layer. It is the ground tissue that is present between both the epidermal layer, and the mesophyll is not differentiated into Palisade parenchyma and spongy parenchyma as in the previous case of dicot leaves. And then the cells are irregularly arranged with intercellular spaces. These cells also have chloroplasts. Then the last layer, which is the vascular bundle. Large number of vascular bundles are present in the case of monocot leaves. Some of them are small and some of them are large. Each vascular bundle is surrounded by a parenchymatous bundle sheath. Vascular bundles are conjoint, collateral and closed. Xylem is present towards the upper epidermis and phloem is present towards the lower epidermis. So these are the features of a monocot leaf. Now coming to the differences between monocot and dicot leaves. The dicot leaf is dorsiventral leaves, which means that the dorsal side and the ventral side are distinguishable, whereas the monocot leaf is isobilateral leaf, which means that both the dorsal side and ventral leaf are looks almost similar. And then the mesophyll is differentiated into palisade and spongy parenchyma in case of dicot leaf, whereas the mesophyll is not differentiated into different parenchymas in the case of monocot leaves. And then further, the differences are the bulliform cells are present in monocot leaves, whereas the bulliform cells are absent in a dicot leaf. So these are the basic differences between a monocot leaf and the dicot leaf. Plant physiology is the subdiscipline of botany that is concerned with the functioning of functioning or the physiology of the plants, whereas the plant anatomy is the study of internal structure of the plants. So this is the basic difference between plant physiology and plant anatomy. Plastids are double membrane bound organelles found in plants and some algae. They are responsible for preparation and storage of food. Plastids are of three types. They are chromoplast, chloroplast, and leucoplast. Now coming to the chloroplast, green plastids containing green pigments called chlorophyll. Chloroplasts are oval-shaped organelles having diameter of 2 to 10 micrometer and then thickness of 1 to 2 micrometer. So when we come to the structure of chloroplast, it has an envelope. Basically, the chloroplast envelope has an outer membrane and an inner membrane, and these two membranes are separated by an intermembrane space. Next comes the stoma, that is the matrix present inside the membrane. It contains DNA and ribosomes and other molecules that are required for protein synthesis. And then comes the thylakoid. It, it consists of thylakoid membrane that encloses a thylakoid lumens. Thylakoids are small discs. They form a stack of disc-like structures called grana. In singular, it is granum. And then these granas are stacks of, these disc stacks, thylakoid stacks, they are interconnected to each other by membranous lamellae called the Fred channels. Now, coming to the functions of chloroplast, basically it helps in photosynthesis, it helps in storage of starch. Synthesis of fatty acids are being done in this chloroplast, and then it also stores lipids, and it also helps in formation of other chloroplasts. Coming to photosynthesis, photo means light, and synthesis means to build. And photosynthesis is a process by which autotrophic organisms like green plants, algae, and chlorophyll containing bacteria utilizes energy from sunlight to synthesize their own food. In this process, carbon dioxide combines with water in presence of light and chlorophyll to form carbohydrates, and oxygen is released as the byproduct. Photosynthesis occurs in green parts of plants such as leaves, stems, and floral buds. Pigments involved in photosynthesis are called photosynthetic pigments. Photosynthetic pigments are of two classes, namely primary pigments and accessory pigments. Chlorophyll A is a primary pigment that traps solar energy and converts into electrical and chemical energy, thus it is called the reaction center. Other pigments such as chlorophyll B and carotenoids come under the class of accessory pigments. They pass on absorbed energy to chlorophyll A molecule. The reaction centers and the accessory pigments are together called as photosystems. Now coming to the role of sunlight in photosynthesis. The entire process of photosynthesis takes place in chloroplast. The structure of chloroplast is such that the light-dependent reaction and light-independent reaction take place at different sites in the organelles. So in the light-dependent reaction was found by Robin Hill in 1939. This reaction takes place in presence of light energy in thylakoid membranes of the chloroplasts. So basically, photosynthetic pigments absorb light energy and convert it into chemical energy, that is ATP and NADPH2. These products of light reaction move out from the thylakoid to the stoma of chloroplast. So that is being depicted in the light-dependent reaction half of in the figure. Then basically ATP is adenosine triphosphate, ADP is adenosine diphosphate, NAD is nicotinamide adenine dinucleotide, and then NADP is nicotinamide adenine dinucleotide phosphate. So this is the gist of light dependent reaction. And coming to light independent reaction, as the name suggests, it is light independent. This is carried out in stoma. So CO2, carbon dioxide, is reduced to carbohydrates with the help of light generated ATP, that is adenosine triphosphate, and NADPH2. This particular light independent reactions are also called as Calvin cycle, whereas the light dependent reaction is called as the Hill cycle. So here the inputs are carbon dioxide from the atmosphere and the ATP and NADPH2, which is produced from the light reaction. So the factors affecting photosynthesis can be of two types. So they can be classified broadly under internal factors and external factors. Then internal factors include pigments, leafage, accumulation of carbohydrates and hormones. And then the external factors include light, carbon dioxide, temperature, water and minerals. Just a key point, just a fact here, the artificial photosynthesis is a method producing renewable energy by the use of sunlight. Indian scientist CNR Rao, who conferred the Bharat Ratna Award, is also working on the similar technology of artificial photosynthesis to produce hydrogen fuel. So that is all about the role of sunlight in photosynthesis. Now we are coming to the next cell organelle, which is mitochondria. Mitochondria are filamentous granular cytoplasmic organelles present in the cells. They were first discovered by Kolliker in 1857 as granular structures in striated muscles. They are also referred to as the powerhouse of cells. So these cell organelles within the eukaryotic cells, they produce adenosine triphosphate, which is ATP, which is a form of energy of the cell. So hence they are called the powerhouse of the cell. They vary in size from 0.5 micrometer to 2.0 micrometers, and they contain 60 to 70% of proteins, and then 25 to 35% of lipids, 5 to 7% of RNAs, and small amount of DNA and minerals. So coming to the structure of mitochondria, basically it has mitochondrial membranes. It consists of two membranes called the inner membrane and the outer membrane. Each membrane is rough about 60 to 70 angstroms thick, and the outer is more smooth and freely permeable to most of the molecules, small molecules. It contains enzymes and protein. It contains enzymes, proteins, and liquids. It has porine molecules, which are proteins, and the inner mitochondrial membrane is semi-permeable membrane, and it regulates the passage of materials in and out of the mitochondria. It is rich in enzymes and carrier of proteins. It contains 80% of the proteins and lipids. The part which we can observe when we cut open a mitochondria is cristae. That is the inner mitochondrial membrane that gives rise to the finger-like projections, and these finger-like projections are called cristae. The cristae increases the surface area of the mitochondria to hold variety of enzymes. And next we see the oxysomes. The inner mitochondrial membrane bear minute, regularly spaced, tennis racket shaped particles known as oxysomes, which are F1 particles. They get involved in ATP synthesis process. The structure of one oxysome is being depicted in the photo, which is in the left bottom. And then we have the mitochondrial matrix. It's a complex mixture of proteins and lipids. The matrix contains enzymes for Krebs cycle, which we'll discuss sooner. And then it has mitochondrial ribosomes and tRNAs and other mitochondrial DNA. So coming to the functions of mitochondria, mitochondria is the main organelle of cell respiration. They produce large number of ATP molecules, so they are also called ATP factory of the cell. It helps the cells to maintain normal concentration of calcium ions, and then it regulates the metabolic activities of the cell. So coming to the respiration, respiration involves exchanges of gases between the organisms and the external environment. Plants obtain oxygen from their environment and release carbon dioxide and water vapor. This exchange of gases is known as external respiration, and then respiration can be classified into two types called the aerobic respiration and anaerobic respiration. Aerobic respiration is a type of cell respiration in which the food is completely oxidized with the help of oxygen, and when we get carbon dioxide, water, and energy as the output. Whereas in anaerobic respiration, the respiration takes place without oxygen, and then the glucose that is the organic food is converted into ethanol or lactic acid. So as you can see here in the equation, the glucose is being converted in aerobic respiration. The glucose is being converted into carbon dioxide and water plus ATP with the presence of oxygen. Whereas in anaerobic respiration, in the equation you can see the glucose is being converted into ethyl alcohol, which is ethanol. And then carbon dioxide and energy we get as the output. The next thing in respiration is the respiratory quotient, which is the ratio of volume of carbon dioxide liberated and to the volume of oxygen consumed during the respiration. So the formula is written here in this slide. Now coming to the stages of aerobic respiration, we have the glucoses, which is nothing but glucose splitting. The breakdown of one molecule of glucose into molecules of pyruvic acid. So this takes place in the cytoplasm of the cell. It is the first step of both aerobic and anaerobic respiration. Next, we have the Krebs cycle. The cycle occurs in mitochondria matrix. At the end of glycosis, two molecules of pyruvic acid enter into mitochondria, and oxidation of pyruvic acid into carbon dioxide and water takes place through the cycle. This is also called as tricarboxylic acid cycle. And then we have the third process, the electron transport chain. This is accomplished through a system of electron carrier complex called the electron transport chain, located on the inner membrane of the mitochondria. NADH2 and FADH2 molecules formed during the glycosis and Krebs cycle are being oxidized to NAD plus and FAD plus to release energy via electrons. The electrons, as they move through the system, release energy which is trapped by ADP and to synthesize ATP. This is called the oxidative phosphorylation. In this, oxygen, the ultimate acceptor of electrons, get reduced to water. Thank you for watching the video. Please do like, share and subscribe the video, and